To understand the problem at hand better, first let us look at the process by which our bodies manufacture proteins from genes and the steps at which we can interfere to produce a variability. The process of formation of proteins, or the central dogma, is surprisingly not so much of a chemical concept but rather an informational one. The process starts at DNA, where the information about the structure of protein is coded in its most primitive form as found in nature. But the DNA is enslaved by its geography, that is, it is only found inside the nucleus. To carry this information, another molecule called messenger RNA is employed. One way to study the function of a particular gene is by silencing it and then studying the resultant cell structure. RNA interference is a technique which is used to knock down genes. Gene-specific small interfering RNA molecules are used to bind with particular messenger RNA to produce the said loss of function or gene knockdown. Imaging techniques try to capture the changes introduced by gene knockdown. Cell painting uses 6 unique dyes, hence staining 6 different locations inside the cell: nucleus, endoplasmic reticulum, actin, nucleolus, mitochondria, and the Golgi apparatus. The composite image is made by combining all the 6 channels. To understand the data fully, we need to understand the experimental setup under which the data is collected. There are 2 main parts to the process. First, the experiment, and then the data point. Cell cultures are created by introducing a cell with appropriate dyes and a reagent. These cultures are created in wells of plate which can hold 384 such wells. There are 4 plates in an experiment. One well can be thought of as a mini test tube. There are 2 imaging sites per well. An image is collected from each of the sites and is downsampled from their original size. The final size of the image is 512 cross 512 cross 6. That is, there are 6 channels per image. The siRNA is the object of interest, which is going to create the biological variability, or on a simpler level, changes which we are going to observe. There are 1108 different siRNA reagents used here in total. The goal is to predict the correct siRNA label using the image data. Now I will highlight 2 important statistical details about the dataset. First, the class distribution, that is the number of samples per class. A higher number for the average number of samples per class is always good. That is, we would also prefer a higher number of images in the training set, but we would not want a high number of classes. This plot shows the distribution of classes sample-wise. For this dataset, the average number of samples comes out to around 30 samples per class, which is not a very good number. Next, we will look at the distribution of pixel values. Plotting the distribution of mean pixel value per image, we would expect it to peak at a particular value and would not want it to spread out. Here we can see the channel-wise distribution of the mean pixel value. The distribution is not good for channel 2 and is towards ideal in channel 5. Various methods and techniques were tried in modeling the problem. Here I will showcase the ones which had a positive impact on the result. The methods and techniques can be divided into these parts. First, let us look at the factors which played a part in choosing the network. Residual networks, or ResNets, exploit the universal approximation theorem, which states that a neural network can asymptotically approximate any continuous function. Here, instead of approximating h of x, we approximate h of x minus x, which we call the residual. This imparts a very interesting property to the structure of the network. To compute the addition of the input with the output, a shortcut connection is made between the two. The benefit of the shortcut connection comes during the backpropagation phase, where extra gradient flows through the shortcut connections. DenseNet builds upon ResNet and takes the idea of shortcut connection to an extreme. Instead of adding the input of the last layer to the output, in DenseNet, the output of all the previous layers is concatenated. This diagram shows one submodule in the structure of the DenseNet. Here we will take a look at the functions which were considered to act as a loss in this problem. First, the softmax loss. Softmax is actually a misnomer because it is actually a cross entropy function which acts on the softmax of the outputs. Softmax acts in two ways. First, it normalizes the output so that each entry can be considered a probability. And second, it smoothens out the output so that it becomes differentiable. The final expression is calculated by summing over for each data point in a batch the value of the loss. Triplet loss operates on the fact that the distance between samples from the same class should be less than the distance between the samples of different class. Here, blue is the anchor, green is the sample from the same class, and red is the sample from a different class. Triplet loss enforces that the distance between blue and green should be less than blue and red. The same idea can be expressed using multiple expressions. Arc margin loss can be motivated using a softmax loss. The point of cloud shows data points without any structure. Softmax loss induces clusters on the point cloud. Now, if we force the magnitude of the points to a particular value, say S, they are equidistant from the origin and we end up with the arc phase loss. Introducing an angular margin of angle M, the cluster of points becomes tighter. Learning rate determines the step size which an algorithm takes. Proper scheduling of the learning rate is crucial for the convergence. The graph shows the evolution of the learning rate with epochs. The red graph shows a stepwise decaying learning rate and the green graph shows a cosine decaying learning rate, which is used in this problem. Label modification techniques can be used to manipulate the topology of class clusters in the embedding space. In pseudo-labeling, in place of labels, pseudo-labels are used, which are nothing but picking the class which has the maximum predicted probability. Pseudo-labeling helps the network to become insensitive to the variations and help induce tighter clusters. Data augmentation is a broad variety of techniques which is used to increase the available training dataset. It also helps the model to become robust by increasing the feature capturing strength of the filters. Mix-up is a technique which generates new points by taking a linear combination of existing data points. On the other hand, cutmix generates new points by using negative and positive masks from existing data points. The structure of the experimental setup can be exploited to make better predictions. This is done using something called linear sum assignment. One physical way to think about linear sum assignment is as follows. You have a 5 cross 4 checkered board and you have to assign 5 pieces on the board such that no two pieces share the same row or column. The yellow squares show one such assignment. The experiments are designed in such a way that no two images share one label on a single plate. There have been various techniques proposed in the recent times to understand the workings of a deep CNN. Here we have used a sophisticated gradient-based visualization technique called Grad-CAM. Grad-CAM helps us answer the question, where is the model looking at? Activation maps are generated for a particular layer. Here, the activation map A1, A2 and AK are generated as an output of layer L. We can think of the class activation as a function of these activation maps. Changes at a spatial location ij in an activation map will produce changes in the class activation. A summation over all such positions in an activation map will give the importance of that activation map on the class activation. The final Grad-CAM visualization is produced by summation of all the activation maps weighed by their importance, as calculated by taking a gradient in the previous step. It is observed that the inclusion of cell type is important for a better prediction. The shape of the input is 512 cross 512 cross 6, which changes to 2208 cross 1 cross 1 after convolution. There are 4 cell types and the information about the cell type is captured by a one-hot vector of dimension 4. It is observed though the arc margin loss is discriminative, it produces an unstable training regime. Therefore, a linear combination of arc margin and softmax is used. LS is label smoothing, CM is cut mix, MU is mix up, CD is cosine decay, and PL is pseudo labeling. Here we can see the performance of the model in terms of the training and the validation accuracy. We can see a massive jump in the validation accuracy after pseudo labeling is introduced. A very informative way to understand the performance of a model is through template and feature vectors. Here X is the model output, which is a vector of size 2212. A template matrix W is pre-multiplied with the model output X to get the probability vector P'. Pi, which is the probability that feature vector X belongs to class I, is directly proportional to the dot product between the template vector Wi and feature vector X. For a better understanding, let us look at a toy example. Here, the red, green and the blue vectors show the template vector. The yellow dot is the end of the feature vector. The probability that the yellow dot belongs to red, green or blue is shown on the right. You can see it change as the yellow dot moves around. From this, we can draw two conclusions. First, that the template vectors should be as far apart as possible. And second, that the feature vectors should be clustered together, preferably around the template vector. You can see in the scatter plot of template vectors that after the introduction of pseudo labeling, the scatter plot becomes much more spread out. Similarly, the scatter plot for feature vectors show that after the introduction of pseudo labeling, the clusters are much more tighter. Finally, to understand what the network has learned, let us look at the Grad-CAM visualizations of the last layer. These activations are overlaid on a particular channel. An important contribution of this work is learning from attention. Suppose we were to answer the question, what is the model looking for? In this case, it is difficult to answer such questions because we don't know the mechanism of actions of the reagent, also the semantic meaning of those actions. But if we trust the model, we can easily flip these questions. Now the problem is we don't know the mechanism of the action, but it can be answered in the form of what is the model looking for? Or in other words, the mechanism of action of the reagent can be understood by looking at the visualizations obtained from the model. This plot gives a glimpse of that idea. For the sample shown before, the reagent pays high attention to channel 4 as compared to other channels. This should be an indication that the reagent is acting on the nucleolus because that is the part captured in channel 4.